Hello, my name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation and Chair and Founder of the World Stem Cell Summit. Thank you for coming to another session of our public day. In the history of World Stem Cell Summits, we had a great meeting back in 2010, and one of our partners was the University of Michigan. It was in Detroit at the Renaissance Center there, and it was there that I was acquainted with Dr. Gary Smith, our next speaker. Dr. Smith is a leading scientist in the field of pluripotent stem cells, embryonic stem cells. He's created stem cell lines that are with the National Institutes of Health registry. I can't think of a better person to explain some of the basics of stem cells as the engine of regenerative medicine, let's all welcome Dr. Smith.
Great. Thank you very much, Bernie, for the kind introduction. And as we get started today, I'd like to thank all of you for taking time out of your busy schedules to participate. And this is really a great day and a great couple days for discussing stem cells, regenerative medicine, and where the science is going. And so today what I'd like to do is talk to you. In the next 30 minutes, we cannot cover all the basics of stem cell biology, but we definitely will cover some of the differences. And before I start, I'd like to acknowledge the team that I work with. It's really their hard work and their dedication, their talents that really give me the opportunity to talk to you about the things that we're doing. So, discussion, what I'd like to talk about is what is a stem cell? Very basic. What are the different types of stem cells? How can stem cells be used? And specifically, we'll talk about their potential use in regenerative medicine, and then also in disease modeling. And when we talk about disease modeling, we'll break it down into discovering how bad genes cause a disease, and then also how these cells can be used for potential therapeutic screening in the future, and I'll give you some examples of that. So it was mentioned earlier today that our bodies naturally have stem cells. And so while stem cell science is a fairly new area of study, the actual importance of stem cells has been around us from the beginning of time. We are losing millions of cells every second, and without a supply of new cells, we would lose our gut, which might not be a bad thing for some of us, but lose our gut, or the lining of our gut, in a few days. We would lose our skin in 3 weeks, and we would lose our red blood cells in about 4 months. So within our bodies, there are stem cells that have specific functions. Some of the important things is trying to figure out how they function and how they work, how they might be kept quiescent, or how they might actually function when they're damaged. So very basic properties. What makes a stem cell a stem cell? And this list can go on, and as our science progresses, we find that there's more descriptions. But very basically, a stem cell is a cell that has the ability to self-renew or replicate and survive for a very, very long period of time. And sometimes this might be indefinitely, like some issues that I'll talk about with human embryonic stem cells. In addition, besides self-replicating, while they replicate, they need to maintain a normal chromosome number. And third, they have to have the ability to develop into a cell with specific functions and characteristics that are different from the cell of origin. So they self-replicate, but they also can become a specialized cell. So one thing I'd like to point out as a learning moment is not all stem cells are the same. There is some stem cell inequality. And I'll use an analogy here. If we look here, all of these are animals, but they're surely not the same. My dog, Grace Kelly, is not the same as a hippopotamus and is not the same as a giraffe, yet they are all animals. The same is true with stem cells. Embryonic stem cells, induced pluripotent stem cells, child, adult stem cells, they are all stem cells, but they are not all equal. So let's start talking about these different types of stem cells. First, embryonic stem cell. And I will say this is the stem cell that I spend most of my time working on. There are controversies around embryonic stem cells, but that's not what today's discussion is about. Embryonic stem cells come from an embryo. In the work that we do, they come from human embryos. The egg is fertilized, it begins to divide, and by about day 5, we have what is called a blastocyst. It has a layer of cells that are green here that will eventually become the placenta, and those that are pink, which would eventually become the fetus. At day 5, these cells, there's about 30 of them, are undifferentiated. There is no difference between those pink cells, one to the other. We can isolate those and make human embryonic stem cells. We also see here an electron micrograph, and my pointer doesn't work, but we actually can see what is called an inner cell mass, or an ICM. That's where the embryonic stem cells come from. The outside cells are those cells we call trophectoderm. So inherent in making a human embryonic stem cell is the need for a human embryo. These human embryos come from one source, from in vitro fertilization. You cannot take an embryo out of a person, and there are very strict regulations, state and federal regulations, that say that we cannot make a human embryonic stem cell from an embryo that is not made for reproductive purposes. So what that means is that we cannot make an embryo to make a stem cell. Every embryo that we use to make an embryonic stem cell comes from an embryo that was made for reproductive purposes. So how does that happen? That's in vitro fertilization. So briefly, as a review here, we know that women get hormones to cause many eggs to develop on the ovary. Maybe 10, 15 eggs develop on the ovary. Those eggs are harvested. Once they are harvested, they are mixed with sperm. This has generally been done for the last 30 years in treating infertility. Day 1, we look for fertilization. Day 2, day 3, day 4, day 5, the embryos are grown. And then it is that blastocyst that has the inner cell mass that we can actually use to make the human embryonic stem cells. Well, a lot of my background is in human embryology, and I'll show you this in a moment. I have, and I still do, in the last 20 to 30 years, treated human infertility from a laboratory perspective. So if we think about in vitro fertilization and how embryos can be obtained to make human embryonic stem cells, there are 2 sources. One is a source that most people know about, and the other is one that most people don't know about. So the first source is a couple who has infertility. They go to a doctor, they get treated for their infertility, they do in vitro fertilization, and they actually generate multiple embryos. In this example, they've made 9 embryos. They use some of those embryos to have a child. They freeze some of those embryos to make children in the future. And in this example, let's say they're very efficient, and they have 7 children, and they have 2 embryos that are remaining that they do not want to continue to build their family. What do they do with those embryos? They have many options. They can donate those embryos to another couple. They can discard those embryos, that's their choice. They can keep those embryos frozen indefinitely, or they can donate those embryos to research, and one of the areas of research is to production of human embryonic stem cells. So this is something when we consent people to donate embryos, all of the options are known to them. We talk to them, we tell them their options, and it is their choice. In fact, it is mandatory that there is a line of demarcation between our laboratory and the healthcare provider. So we cannot be the ones that actually talk to them about the options. The healthcare provider provides them the options. And if they want more information about donating embryos to stem cell research, they can contact us. The other group that most people don't think about are couples who have already had a child or know they have a family history of a devastating disease. And so there are many of these diseases that we call monogenic diseases, known to be caused by one single gene mutation or a few gene mutations. So you can think about cystic fibrosis, some of the muscular dystrophies, Tay-Sachs, SMA. Many of these different diseases are monogenic diseases. And so many of these couples, as I said, either know they have a family history or have already had a child that's been affected and may have passed away from the disease. They no longer want to have another child to go through that process. They want to have a healthy child. So what can be done is they will do in vitro fertilization not because they have infertility, but because they want to have their embryos tested in the laboratory as to whether or not they carry the gene mutation for the disease or not. If they don't have the genetics for the disease, they will transfer the embryos and have a child that doesn't carry that disease in their family any longer. In the past, all of those embryos that were found to be affected or that had the mutation in the gene were discarded. What we've done at the University of Michigan is made a program to where these families can donate those affected embryos to us. We make embryonic stem cells that have these mutations and then distribute them to researchers across the world to study the disease. And in fact, over the last 5 to 10 years, this is actually something that many families now ask us for because they recognize that, well, they have built a way in their family to remove Tay-Sachs, to remove cystic fibrosis, to remove Fragile X syndrome. They still have relatives with these problems. And so they are actually not only having a child without the disease, but now also contributing to research across the world towards understanding the disease. So a little bit more about the embryo analysis. So this is something we do quite commonly now, where we can take one cell out of a day 3 embryo called a blastomere, or we can take 5 to 6 cells out of a blastocyst, and then we analyze it. We can freeze the embryo, get the analysis back, and know which embryos are normal and which ones are abnormal. And as it says here, there have been over 190 genetic diseases for which embryo analysis at a genetic level can be done and circumvented passing that disease on to the children of the family. And I think the other thing I want to emphasize that most people don't recognize is those affected embryos in the past, their destiny was discard. Now they have an ability to donate to actually help us understand these different diseases. The other thing I want to point out now is that as a scientist, I think we have a love for discovery. We're very passionate about the things that we do. We want to make progress, but progress takes time. And it would be great if we could all hit home runs, but it actually takes some of those RBIs and some of that building on what everybody can contribute to make advances. And so many times I have people asking me, why haven't we cured cystic fibrosis? Why haven't we cured Tay-Sachs? You've been working on this for 10 years. And I would say that good science takes time. So it's been mentioned recently about induced pluripotent stem cells. Very, very important. I was at the meeting when Dr. Yamanaka announced their ability to transform a skin cell into a pluripotent stem cell. And I think this is a perfect example of how very basic research allowed an advancement. So understanding what were the transcription factors in an embryonic stem cell that made it pluripotent is what gave rise to the ability of actually reprogramming fibroblasts to become pluripotent. So there are some great advantages of iPS cells, and even though I work primarily with human ES cells, my true belief is that we need to push both areas of science forward. It's too early in science to make a decision as to which will give the best results in the future. The last is what we usually call adult stem cells. Actually going through this presentation and thinking about a more general population, I thought, really adult is not the correct terminology. It's not like children don't have these stem cells, they do. So I've termed them here children/adult stem cells. These are what we call multipotent. They can't become every cell of the human body. A human embryonic stem cell is pluripotent and can become every cell of the human body. Some examples here are heart cells, bone marrow cells, brain cells. Some things to think about with regards to these types of stem cells is that they are usually very rare. They are difficult to identify. They can be very difficult to isolate. They may have limited divisions, and they may have DNA stability issues. Again, with that said, there's been some great progress in using adult, child, stem cells. An example of that is bone marrow transplantations. So now I'm going to talk a little bit about why study human embryonic stem cells. I've already mentioned it is controversial, and I know that. But there are biological reasons. Human embryonic stem cells are the gold standard. And if you go to the basics of biology, these cells give rise to every cell of the human body. And therefore, inherent in what they are is to become different cells of the body. Induced pluripotent stem cells from skin, it has been shown that they have developmental memory. There may be experiments or things that they may be used for where that does not matter. And again, this is why I feel we need to study both. Resource ethical issue. We've talked about the controversy of human ES cells, where they come from. You can't make a human ES cell without using a human embryo. But I would say that from a resource perspective, every embryo we use to make an embryonic stem cell would otherwise have been discarded. Therefore, we feel that it would almost be unethical to not put forth this option for people to donate their embryos to try to help the living and help those that have diseases. Last thing I'd say is I have personal reasons. I was raised in a family of firemen. My grandfather was a fireman, my dad was a fireman, my uncle was a fireman, my brothers are firemen. They're all service-oriented people. And as I became a scientist, that's what I wanted to focus on, doing science that was service-oriented. And in fact, that's why I first started in the area of assisted reproductive technologies, helping families that were infertile build their families. And it's something I still do today. I did it at the University of Chicago for many years. I do it at the University of Michigan still. But there was a time 4, 5, 6 years ago when I asked myself, is there a way I could use my skills, my science, my technologies to help beyond infertility? And my answer was to produce disease-specific human embryonic stem cell lines. And I'll get back to the use of those in a few moments. So the promise of human embryonic stem cell and any type of stem cell research can really be taken down to 2 levels. The first level is regenerative medicine, taking pluripotent cells, stem cells, converting them into bone marrow cells, nerve cells, beta islet cells, heart cells, and treating a disease, regenerative medicine. The other area that we focus on primarily in my laboratory is actually modeling diseases, using these pluripotent stem cells to understand how a cell goes from a pluripotent stem cell to a neuron, from a pluripotent stem cell to a heart cell. What are the mechanisms of normal development, or what are the mechanisms of abnormal development? With that, we feel that we're going to be able to understand developmental abnormalities, developmental birth defects, as well as diseases that are late onset, primarily monogenic diseases, not multivariate diseases. In addition, we can use these pluripotent stem cells for screening new or repurposed therapeutics, and I'll give some examples of that later. The other thing I want to point out, and I think it's an important issue, not just for scientists, but also for the general population, is to balance the hype and the hope. There is no doubt that stem cells hold great hope for the future of medicine. It will take good time, studies, and dedication to get there. With that said, we have to be careful about overhyping it. There is nothing that will cause something not to move forward than bad data. Good data will make things go forward and will make them have a strong bedrock in the area of medicine. So when I think about regenerative medicine and stem cells being an engine for regenerative medicine, it comes down to very simple issues of balancing, promise, and caution. And I'll give you an example of something that I did an interview about a year ago. And this was for PBR, public radio. And I was asked about Gordie Howe, Mr. Hockey, being from the University of Michigan, Detroit, very, very famous person up in the colder country. And Gordie Howe had gone to Mexico, and he had had a stroke, and he went to Mexico, and he had a stem cell therapy. And it was being reported that he had a great recovery. And as many of you probably know, Gordie Howe has now passed away. But when I was interviewed, and the interview is here in blue if you want to listen to it at some point, the thing that I tried to emphasize was clinical trials are a must. Clinical trials take time, but they are a must. They tell us about safety, they tell us about efficacy, does it work? It's not anecdotal. It allows peer review and allows FDA approval. All of these things are very, very important if stem cells are going to be the engine for regenerative medicine. A few things I want to bring up as limitations of stem cells for regenerative medicine and some caution. Cord blood, child, adult stem cells do have a limited ability to proliferate and to become all types of body cells. That's not true with human embryonic stem cells or iPS cells. In addition, if you put an adult stem cell in the wrong area, a place where it normally doesn't occur, it does not necessarily mean it's going to develop normally. It could develop abnormally, and there are evidence of that. So that has to be, even though they are self cells, they still can have abnormal development. Using self stem cells for genetic diseases or regenerative medicine of genetic diseases is really almost illogical. To say that I've got type 1 diabetes or I've got cystic fibrosis and I'm going to take one of my own fibroblasts, reprogram it, and make it a nerve cell to treat my disease, if it's caused by a genetic defect, we have just made a cell with that same genetic defect. So those are important things to remember. The other thing, when we think about pluripotent stem cells as being important for regenerative medicine, we're not talking about them as a pluripotent stem cell. We're talking about them as a pluripotent stem cell that's been made into a specialized terminal cell. So making a heart cell to treat a heart attack. And this is very important because if one was to think about just transferring pluripotent stem cells, we know they have the ability to form tumors and to form teratogens. So very, very important to know these subtleties. In the last bit of my talk, I'd like to talk about this modeling or disease modeling. And as I've mentioned, this really makes sense for single gene diseases, where we know a gene problem causes an abnormal cell or organ system. But what we don't know is how that disease develops and how it progresses. That's really our black box here. However, if we can make human embryonic stem cells or pluripotent stem cells, iPS cells, for these diseases, now we have a way of looking at how they develop into abnormal cells, tissues, and organs. And as a scientist, one thing I think holds true: knowledge is good, lack of knowledge is bad. So the more knowledge we have, the better off we're going to be in trying to fight some of these diseases. The other thing I want to bring up here is that it's a quote by Helen Keller: "Alone we can do so little, together we can do so much." And one of the very important concepts in our program, M Stem Cell Laboratories at the University of Michigan, is not that we do everything. We do what we're good at. We use our embryology skills to make very good and efficient human embryonic stem cell lines. As soon as we make those lines, it's not the best use to stay in our hands. It's the best use to go into the disease-specific researcher. So when we make a line, we will reach out to any researcher we can find at the University of Michigan, as well as other researchers worldwide. So our goals are to derive novel disease-specific human embryonic stem cells and get them out into the worldwide research community to accelerate our understanding of human diseases and to develop new treatments and cures for these novel diseases using these novel disease-specific human embryonic stem cell lines. We support good, rigorous science, and we believe this is going to support development of treatments. Lastly, we feel it's very important that we spend time engaging and educating not just the general population, but actually the scientific population. So here are some of the different stem cell lines that we've derived over the last few years. These are all disease-specific. These are all embryos donated to the University of Michigan under informed consent, IRB approval. Every stem cell line we make, we send all of our information to the National Institutes of Health. And I'm very happy to say every stem cell line that we have produced has been accepted on the registry of the National Institutes of Health. So you can see there's some for aniridia, blindness. There are some for Huntington's disease. There are some for SCA3, spinal cerebellar ataxia. We just made one for ALS, frontotemporal dementia. Again, any disease caused by a single gene mutation, we can actually obtain embryos that are being tested for, that are affected, that would have been discarded, and make these disease-specific human embryonic stem cell lines. The other thing I'll mention here is that you can see some of these in gray. One of the neat things that we're starting to do is have multiple lines of stem cells from different embryos. And some of them are from the same family, and some of them are from different families. So if we think about science and wanting model systems, we want things that are genetically similar, but some that are genetically not genetically abnormal, but genetically dissimilar. So we can start testing, does this happen within one family, or does this happen in multiple families? In addition, we make control stem cell lines, and again, every line we've made is on the registry, and any researcher in the United States as well as around the world can obtain these lines from the University of Michigan. Let me give you some examples of some of the things that we've done with these stem cell lines. The first line I'll talk about was one that was for hypertrophic cardiomyopathy. This is a lethal disease. You know this, many young athletes will die of a heart attack without ever knowing they had a problem. This is a family that had a mutation in what is called myosin-binding protein C3, causing hypertrophic cardiomyopathy. We were able to make a human embryonic stem cell line from this embryo, make heart cells from the stem cell lines, and demonstrate that they actually mirrored the disease in a petri dish. Adrenoleukodystrophy. This is a disease that's, again, lethal. Most boys die of this by the age of 19. We've been able to make a human embryonic stem cell line from an embryo that had the mutation causing this disease, getting it to researchers across the country, and they're starting to understand how this disease develops, how it interacts with other cells, and bringing about the disease, such as looking at its interaction with the immune system. Fragile X syndrome, causing intellectual disabilities in young men who have this Fragile X mutation. We're actually able to make neurons from these embryonic stem cells and study the disease in a petri dish. If you could please click on this slide, please. This actually shows the heart cells that came from a normal human embryonic stem cell line, as well as the heart cells that came from our disease-specific hypertrophic cardiomyopathy stem cell line. So these are the heart cells. It takes about 30 days to make them from the stem cell. But you can see that the heart cells from the stem cells that had the mutation beat abnormally and with much more abnormalities compared to the controls. And this allows us to say, now we have a model to study the disease in a petri dish. Now we can start doing things like screening of therapeutics. The last thing I'd like to do is acknowledge funding for this project. Human embryonic stem cells, because of its controversial nature, cannot be funded by the National Institutes of Health or with federal funds. Therefore, all of our work on deriving new human embryonic stem cells is funded by private, foundation, or local funds. From the university, there's been a commitment to this project, one of the few universities in the country committed to this project. One individual I'd like to mention, Mr. Alfred Taubman, who has passed away, actually a Michigan man, but also from Palm Beach in Florida. And he was one of the very important founders of this project. Presidents of the university, the executive vice presidents of our health system, the dean of the medical school, Dr. Eva Feldman, and Dr. Tim Johnson. The last thing I do is leave this slide up for you. This is an information slide. If you or you know of anybody who might be thinking about pre-implantation genetic diagnosis of embryos for a familial disease, instead of discarding their affected embryos, we have the ability to accept those embryos and very efficiently make embryonic stem cell lines that then can go on the NIH registry and be given out to researchers across the country and the world. Thank you.